Well, hello, everyone, and thank you for attending today's discussion. A few housekeeping items before we get started. This conversation is being recorded and will be sent to your email in the days following the event. If you have any questions during the event, you can submit them through the Q&A module at the bottom. Don't be shy in waiting to the end of the presentation, as I'll be fielding these questions throughout the discussion. So if you have a question, please put it in the module so we can get it answered for you. In addition, we'll be asking a few polling questions during our discussion, so your participation in those would be greatly appreciated. By way of background, my name is David Wicks. I'm Vice President of Listings here at Nasdaq, and I'll be your moderator today. And today we're very fortunate to be joined by Dr. Jim Herman and Dr. Hannah Jones for this exciting conversation. Thank you both for being here and lending your expertise. So before we get started, let's introduce them. First, I'll start with Dr. Jim Herman, Vice President of Toxicology at Certara. Jim, would you like to introduce yourself to the audience, please?
Sure, thank you, David. So my name is Jim Herman. I'm a board certified toxicologist. I've been in pharmaceutical R&D for about 35 years now. Some of it at large pharma, some of it in consulting groups, and I currently lead the toxicology group at Certara, and we provide strategic, operational, and tactical support to biotech and pharmaceutical companies. We help clients achieve their drug development goals by designing non-clinical toxicology programs that meet regulatory expectations, assist the client in finding contract research organizations to conduct the required studies. We manage and review the results of those studies, and we prepare the non-clinical content of various regulatory submissions. So thank you, and it's a pleasure to be here this morning.
Thanks, Jim. So also joining us, we have Dr. Hannah Jones. She is Senior Vice President of PBPK Consulting. Hannah, can you introduce yourself to the audience?
Hi, so my name's Hannah Jones. I'm a pharmacokineticist. I have about 20 years experience in kind of large pharmaceutical companies. I currently lead a consultancy team at Certara that provides support to biotech and pharmaceutical companies. In my team, we use the Simcyp simulator, an in silico biosimulation tool to predict the exposure of drugs or drug candidates in animals and humans with limited data inputs. For example, we predict the exposure or the pharmacokinetics of drugs before going into clinical trials. We predict how drugs interact with each other and how exposure may vary in different populations.
Great. So let's jump in. Historically, the FDA has required drug discovery and development programs to include animal testing. And this requirement ended with the 2022 passage of the FDA Modernization Act. And this change has really opened the door for expanded use of alternatives to animal testing, including biosimulation, organ on a chip, and cell-based assays. In this panel discussion, we'll discuss how scientists can use innovative leading-edge technologies more fully in future drug discovery and development strategies. So I think a good place to start today's discussion is by first understanding the FDA's Modernization Act. So, Hannah, what is the FDA Modernization Act?
So the FDA Modernization Act is part of the Omnibus Bill signed by President Biden just recently in December 2022. The new law eliminates an 80-year-old mandate requiring drug developers to conduct animal tests before human trials, and instead allows them to use non-animal and human relevant methods. The bottom line is really that drug developers in the US now have the option that can result in less reliance on animal testing. They have the option to choose the most appropriate method for their application much earlier in drug discovery, rather than being necessarily forced down an animal route. And this is really encouraging. And during this meeting, we'll talk about some of the alternatives to animal testing. We'll need these alternatives. We will need to prove that they are predictive before moving away from in vivo studies. And this will obviously take some time, but this is certainly a really good first step.
And what is the current status regarding use of animal studies in drug discovery and development?
Well, I'm sure as many of our audience know, animal studies play a key role in drug discovery and development. And the purpose of these studies is to really help identify the best drug candidates to bring forward and support the 3 phases of clinical development. And animal studies not only benefit the development of human pharmaceuticals, but they're also very important for veterinary medicine. So a lot of the drugs that initially may have been targeted for human use also find use in the veterinary space. So both humans and animals development, or they benefit from this testing. So positive results in pharmacology studies in animal models of disease can indicate that a drug candidate has the potential to be efficacious in human disease. Non-clinical pharmacokinetics and drug metabolism studies can predict how a drug candidate will be absorbed, distributed throughout the body, metabolized, and excreted in humans. And non-clinical safety or toxicology studies can identify hazards that may be relevant to humans. And together, the results of these non-clinical studies can help define doses and exposures that can be safely studied in clinical trials. And I'll have a little bit more to say about animal use today later in the webinar.
Great. Thank you, Jim and Hannah. So that leads us to our first polling question. And I would just also remind, feel free to put any questions that you have in the Q&A function. But our first polling question is, are you actively trying to incorporate alternatives to animal testing in your drug development program? So you just have to pick one answer, yes or no. And we'll leave that open for a few seconds. And let's see if we can share some of the results. So Jim, Hannah, 62% yes, 38% no. Surprised by the results, or is this what you expected?
This is probably about what I expected. Jim, sorry.
Yeah, I was going to say the same thing. It's not too surprising because there are some well-established alternatives in silico and in vitro that people have been using and employing. But I still think that there is a lot more opportunity to push that bar even higher, the yes bar even higher, and reduce the number of animal studies that will be required in the future.
Yeah. And I think people need to kind of build confidence as well in the translation of these approaches. And I think that's why there's still a number of no's there.
Okay. So Hannah, how is animal testing used in other industries? Can you talk a little bit about that?
Yeah, so there's already been a lot of precedent for minimizing animal testing, particularly in the EU. And this slide here kind of highlights the cosmetic industry on the left and the pharmaceutical industry on the right. In the cosmetic industry, the EU has had a long history around banning animal testing, beginning in 1993 and culminating in both a testing and a marketing ban that prohibits animal testing on finished cosmetic products. The US FDA does not currently prohibit animal testing in the cosmetic industry, although it does really encourage alternative approaches. In a similar way, in 2019, the US Environmental Protection Agency, the EPA, announced that it would reduce and eventually aim to eliminate testing in animals. And then on the right here on the slide, in the pharma industry, in Europe, the EMA announced a similar approach to the one in the FDA's Modernization Act 18 months ago, and has been investing in and encouraging these alternative methods. So it is getting more established in other industries and across the world.
Great, thank you, Hannah. And Jim, can you talk a little bit about what are the 3 R's?
Yes. So the 3 R's simply are, with respect to animal usage, replacement, reduction, and refinement. And this concept was first developed as, the principles were first developed as early as 1959 to provide a framework for better stewardship of animal research or animal use in research. And the goal of the 3 R's is to find alternative to animal testing, i.e. replacing them, to optimize the amount of information that we get and get it from fewer animals, reducing their use, and to adopt methods that alleviate any distress within the animals, or refining the techniques that are being used. And these guiding principles aim to improve both the quality of the science and animal welfare when the use of animals is unavoidable. One of the major contributions that resulted in a substantial reduction in animal use was the adoption of globally harmonized guidelines and regulatory authority requirements for the non-clinical studies that had to be done to support human medicine development. And really this effort started in about 1990, when the International, what was then Conference of Harmonization, came together, or ICH for short. And basically what this was a group of regulators and drug developers from around the world who got together to discuss and align the scientific and technical aspects of drug development. Prior to 1990, regulatory authority requirements for studies differed by region. And so in some cases, you would have to do 3 or 4 different studies, say reproductive toxicology studies, to support registration worldwide, because the US might require one type of study design, Japan might require another. And, you know, as you can imagine, that greatly contributed to the cost of animal use in drug development. However, with the advent of ICH, study designs have been largely standardized and reduced that unnecessary duplication of effort. And in fact, some studies, such as the acute LD50 studies, which utilized many animals, have been eliminated completely. And I will say that these efforts are still ongoing. In fact, in August of last year, ICH updated a guidance document to allow sponsors to possibly eliminate the need to conduct lifetime rodent carcinogenicity studies. So ethically and economically, it makes sense that the pharmaceutical industry continues to look for opportunities to implement the 3 R's. And certainly with the passage of the Act, it gives more flexibility to FDA to consider these alternative approaches.
Thanks, Jim. So Hannah, in the Act, they refer to alternatives to animal testing. Can you talk a little bit about what are these different approaches and how are they being used currently?
So I think the move away from animal testing is really becoming feasible, like you say, due to the development of these sophisticated alternatives to lab animals. And these alternatives really fall into 3 key categories: the in vitro approaches, organ on a chip approaches, and in silico approaches. Starting off with the in vitro approaches, these are widely used particularly in the area of pharmacokinetics, to predict absorption, distribution, and metabolism of drugs in humans. For example, human hepatocytes can be used. They contain like the key drug metabolizing enzymes and are routinely used to predict the metabolism of drugs in humans. In vitro assays are also used to understand the potency and pharmacology of drugs, and also as an early toxicity screen and to support interpretation of in vivo toxicity data. So for example, target binding assays to understand the pharmacology modulation, and Hurg assays, for example, to understand cardiovascular toxicity are routinely used. And considerable progress has been achieved in making these types of assays available and deployable in a user-friendly, aligned fashion across the industry. And moving to the kind of so-called organ-on-a-chips or microphysiological systems, these began to be used more frequently during COVID-19 drug and vaccine testing. And they reportedly helped researchers to better understand how the SARS-CoV-2, the virus that causes COVID-19, interacts with human organs and elicits an immune response. Using human cells and engineered structures, these approaches create an environment that mimics or kind of models the function of organs and in some cases, multiple organs and how they actually interact together. These systems are not currently being used routinely, but over time have the potential to be used in many settings in lieu of animal studies. For example, they could have an application in testing drug efficacy in genetically diverse human populations using human genetic material. And then finally, the area that I'm pretty passionate about is the sophisticated biosimulation modeling techniques. They use in silico computer-based testing with virtual patients. It's fast and relatively inexpensive and reveals really rich information about how a drug would perform and how to best design a trial before the drug is ever tested in patients. So biosimulation also offers the flexibility of computer-based testing, allowing developers to optimize trial design and dosing for different patient populations. So overall, these in vitro and in silico approaches are frequently used in the area of pharmacokinetics, followed by the pharmacology and efficacy space, and then to a lesser extent in the toxicology space. It's still early for some of these technologies, I think, many of which still need to be refined and standardized and validated. Like I mentioned, that some of these in vitro techniques are quite well validated and established across the pharmaceutical industry and academia, but some of these organ-on-a-chip technologies and newer ones are less well established at the moment. And once they get more established, we'll be able to kind of use them and validate them, and they can have an application more routinely in drug development. However, I do think the momentum is really building for these approaches, which could ultimately obviously help speed up drug development, improve outcomes for patients, and really reduce, like we talked about, the burden on lab animals.
Yeah. And Hannah and Jim, before we go to the poll questions, we do have some questions from the audience that are relative to this slide, so I'd like to take those. The first one says, can you talk a little bit about use of data from organ-on-a-chip models and how reliable they are considered by the industry, quote, investor confidence, and are likely to be viewed by the FDA?
Do you want to take a stab at this, Jim? Or?
Sure. Well, I think at this point, you know, as Hannah mentioned, it is an emerging technology. And what we have to keep in mind to really make it useful is there has to be a lot more study and understanding about how predictive and how validated these technologies can be, and can we have confidence that they will mimic an in vivo situation, be it in animals or people. And clearly we're not there yet. There's a lot of work that has to be done to do that. And because of that, I think regulators are going to be highly skeptical of relying on those solely. But employing these techniques now may allow drug developers to actually design better in vivo experiments and design better clinical trials based on some of the data we get from these chips. So, you know, they're a long way away from what I would call commercial application. But I think, you know, it's going to take some time and effort to get there. And I certainly hope that people invest that time and effort, because I think the payoff could be very, very large in the end, both in terms of saving time and costs.
Great, thank you, Jim. And a follow-up question. Is it possible to use organ on a chip to evaluate response of an investigational product and compare to approved biologics, for instance, for the treatment of asthma using the lung model? You didn't think this was going to be easy, did you? I can repeat that if you'd like.
Yeah,
Yeah,
can you
just-
repeat it?
So it says, is it possible to use the organ on a chip to evaluate response of an investigational product and compare to approved biologics, for instance, for treatment of asthma using the lung model?
So I think that's potentially where some of the organ on a chip methodologies can be used to compare different therapeutics against each other. I think what's lacking is a formal understanding of how these organ on a chip approaches translate from in vitro to the in vivo setting. So perhaps in the in vitro setting, a kind of comparison approach of different candidates or different modalities could be a way of using them quite efficiently.
Yeah, and I would say in this, for that specific question, it's really going to, when you talk about organ on a chip and you say lung, you have to really define what cell types you're talking about. Are you talking about the epithelial lining cells? Are you talking about the smooth muscle cells that could contribute to asthma? Are you talking about, are there going to be inflammatory cells that could also contribute to the condition? So again, I think there's lots of factors that you have to take into account before you can say, that these models are going to be useful or predictive. Certainly, you could compare an investigational drug with a known drug, but we have to be careful not to overinterpret, because even though you can link these chips together and circulate cell media through them, such that if metabolism happens in the liver, the lungs or kidneys or muscle could be exposed to the parent and those metabolites. There's still a lot we don't know about how relevant and how representative that is to the in vivo situation. So I think that's, again, it's in its infancy, and it's going to take some time and effort to be able to really develop some confidence in that.
Great. Thank you, Jim. So let's go back to the poll question for our audience. So we're going to throw up the second poll question. Which of these alternative methods are you using? And note that you can select more than one answer. And we'll give that a few seconds for our audience to vote. And let's put up the results. So, Jim, Hannah, cell-based assays leading by biosimulation, surprising?
Yeah, I'm not surprised, to be honest, because like I said, I do feel that cell-based assays, particularly in the PK and PD space, have been used for a long time and are very well established. And often we use those cell-based assays in models to simulate exposure or pharmacological effects in clinic. And so potentially they're not always used in models. So I'm not surprised that biosimulation is slightly lower than cell-based assays, and organ on chip is the lowest, because obviously we've talked about how some of those approaches are not fully established yet.
And I guess I would agree with Hannah, and I think the one thing that surprises me a little bit is that there are 11% of the respondents are using cells or organs on a chip, which I think is, to me, very encouraging. So people are exploring the possibility and using them, and I'd like to see that number go higher.
Fair enough. And let's do one more poll question. So this is our third question. What are your biggest concerns about using alternatives to animal testing? And again, please note you can select more than one answer. We'll give that a couple of seconds for people to click on that. And if we want to show the results, please. All right, so lack of knowledge and concern about regulatory acceptance are the major concerns. No surprise there, I assume.
No.
Absolutely not. I think, when we think about who our customers are in pharmaceutical development, obviously there's the patient, and that's first and foremost. That's why we do what we do. But a secondary, and in some cases primary customer is really the regulatory authorities. And you have to be able to convince them that the approaches that you're taking are both valid and will inform on the safe human use of a compound. So I guess these don't surprise me at all, and it really reflects the infancy of how far some of these alternatives have advanced so far. They're just very, very early on.
All right, let's take maybe one or two more audience questions, and then we'll go back to the slides. But one question came in: is organs on a chip something that is used in QSP models, for example?
I think they can be in some cases. Primarily, I think around comparing different molecules, for example, against a competitor molecule or a molecule that has been established in clinic already. And also just to understand a little bit the pharmacology in more detail and how it might act at more of an organ level, and to understand some of the systems parameters related to these models, for example, how the target is turned over and things like that. But again, it is really in its infancy, and these types of models use much more the traditional in vitro techniques, but this will change over time and allow us to kind of explore, rather than at a cellular level, more at an organ level.
All right, thank you for that. Another question from the audience: some ex-US regulatory agencies seem reluctant or slow to adopt new modalities of testing. How do you see that playing out compared to the FDA?
I think the FDA has taken a good first step here, and I think some ex-US regulators are also on board with taking that first step, but they're going to be very cautious because their remit is the protection of the patient, and so again, I think it's going to take some time to convince them that some of these models or alternatives will be useful in the future. But I think, again, to me, I look at this as, if you think back to, in the early 1970s when the Ames assay was first introduced to look at mutagenicity, it took a while for regulators to become comfortable with that. And then later on, when in silico methods were developed to predict mutagenicity, it took a while for regulators to accept those. So it's really going to take the ability to develop concordance between what we're seeing in these alternative methods and what we may expect to see either in animals or in humans.
Thanks, Jim. And Hannah, this is directed towards you. Could you comment on in silico PK/PD analysis for gastrointestinal drug administration modeling?
So perhaps if I've got some slides that kind of summarize some of this, and
Okay, so we can hold
cover
off
that there.
Yeah. And maybe one more question, because we are getting a lot of questions, so I do want to try to get to as many audience questions. Can either of you please comment on the NASA microorgan device technology?
Can you repeat the question?
Can you comment on the NASA microorgan device technology?
I am not familiar with that, sorry.
No, nor am
Okay.
I.
Okay. All right. Well, Hannah, let's switch gears a little bit and talk a little bit about biosimulation, PB, PK, and QSP techniques. So let's start. What are they? How can PB, PK, QSP support the 3 Rs? And can you provide some examples of the kinds of things you can do? So a lot of questions there.
Okay, so in the past, when it comes to PK or pharmacokinetics, exposure prediction, the pharmaceutical companies have been really dependent on animal data to predict exposure of drugs in the body, using allometric scaling techniques as shown on the left, where you measure the particular parameter in vivo studies, plot it against body weight, and then predict what happens in human. In contrast, we now have kind of biosimulation approaches that use computer-based models of biological systems to predict how the body affects the drug and how the drug affects the body. And these approaches are routinely used in the industry now and can reduce the reliance on animal testing. So, for example, these physiologically based pharmacokinetic platforms, or PBPK platforms like SimCip, are large, complex models that capture the transport of the drug in the body and their absorption, distribution, metabolism, and excretion with multiple advanced tissue models. These models are defined by a number of systems parameters like blood flow and enzyme abundance, and they generate virtual individuals based on these systems parameters and the way these parameters co-vary with each other. They generate virtual individuals, which allows us to simulate in populations. And then the user then incorporates readily available drug-specific lab data, in vitro data, to predict the exposure in human populations and also in preclinical species. So we routinely use these models in the discovery setting to predict human exposure with minimal animal data. And really, it's a learning and confirm cycle where the in vitro to in vivo prediction approach is validated with some limited preclinical in vivo data before performing the simulations in human. And then these approaches can be viewed currently, I would say, as a supplement to, rather than actually necessarily a complete replacement for animal testing, but they do have the potential to be used in lieu of animal testing altogether in time as we build more confidence in these different approaches and the mechanisms that determine human pharmacokinetics. So they offer advantages over other techniques because they're mechanistic and they don't require in vivo data across multiple species like some of the kind of older approaches do. I think as well as predicting human pharmacokinetics or human exposure, these methodologies can be used to kind of screen out or rank potential drug candidates against each other to prioritize which candidates to take into human, and also to prioritize formulation efforts, so therefore reducing the reliance on in vivo data. So one of the things that in vivo studies are used a lot for is dogs are used to compare the effect of different formulations on drug exposure. And we have examples where modeling using these types of approaches was used to reduce the reliance on in vivo dog data in this case. For example, we've used modeling to prioritize different extended release formulations to optimize a particular PK profile in human. And in a retrospective example that we're aware of, this was kind of more predictive than scaling directly from in vivo data. So just going back to that question around gastrointestinal absorption, within these PBPK models, we have a detailed intestinal model that allows us to predict how a drug is absorbed as it goes through the GI tract. For example, we can predict how it goes in and out of solution as a function of some of its phys-chem properties and some of the systems parameters within the model, and then we can predict how it is absorbed as a function of its permeability. So depending on the properties, we can really get into the nitty-gritty of kind of GI absorption and predicting potentially exposure for the compounds that we're intending to be slowly absorbed or quickly absorbed, or we want to keep the drug in the GI tract because that's where the pharmacology target is, and things like that. So we have the option here to address quite a lot of GI questions with the model. The other space that I think is very important in terms of modeling is in the pharmacology and efficacy space. So in this setting, in vivo testing in animals is used to understand the relationship between dose, exposure, and effect, with really the objective of translating these effects into human. And so we have a graph here, for example, where we look at kind of tumor inhibition over time in a mouse with different doses of compound. Obviously, these can be quite extensive studies. However, as we know, we're not trying to treat mice. Humans are not large mice. We're trying to treat humans. And there are quite specific species effects which can limit the confidence in translating animal data to human. And so alternatively, we do have what we call quantitative systems pharmacology, or QSP models, that can be used in combination with limited in vivo data. And these types of approaches have led to the reduction in animal testing in this space. And essentially what you're doing here is creating a biological map to understand, or based on the understanding of the biology of a particular disease, and you do this with mathematical equations. So these models are use knowledge of the, maybe for example, things like information around the target, for example, expression levels or endogenous ligand concentrations, internalization, turnover rates, and things like that, together with information about the compound or the drug that has been determined in vitro, to predict target coverage in blood and particular tissues of interest, and also in many cases efficacy. And they can be used together with the PBPK models, the pharmacokinetic models, to actually then translate that to a dose and obviously reduce the number of animal studies. In particular, we have many examples where these types of models have been used to validate a target or predict human dose or optimize a dosing regimen to kind of maximize the therapeutic index, i.e. maximize efficacy and reduce the side effects. And obviously by using models in combination with some limited in vivo data, and we can reduce quite significantly the level of animal usage. So I think these two areas are really growing areas in the pharmaceutical industry, and are going to lead to kind of reduced animal testing over time.
Thanks, Hannah. And Jim, before I ask you for your perspective on toxicology, I do want to take some more audience questions just because we do have a lot, and I want to try to get to some more of these. So first question, is the organs representative of individual, quote, biological variables?
So in these models, what we have is different tissues, and the tissues are kind of defined by maybe, for example, a blood flow, a weight, etc. And we can actually model the distribution of drugs across the membrane of those tissues. And in many cases, drugs move passively across membrane barriers, and this can be predicted from physicochemical data or in vitro data around permeability or protein binding. But in some cases, transporters are involved, and so we have in vitro assays that allow us to test whether a drug is a substrate for a particular transporter, and then we can include those features in the model. So as much as the biology is understood, these models characterize the membrane barriers between tissues and cells.
Yeah, and in
Uh,
terms of organs on the chip, I think one of the things we have to remember is that cell function is all about it's not only about the cell itself, but it's also about the context or the surrounding tissue that it's in, and where it receives inputs from other tissues. So, I think we have to take that into consideration. As well when we're interpreting information from these and we're designing these organs on chips. We have to take into account that the context of that cell in vivo and see if we can replicate it as much as possible in the in vitro setting. And I know there's been, I don't know that it's happening necessarily with organ on the chips, but I know there have been co-culture experiments done before, for instance, where hepatocytes are co-cultured with fibroblasts and Kupffer cells. And that gives you a much more representative picture of the in vivo situation. Now, whether or not that can be adapted to, the organ on the chip technology, I think that's something that needs to be considered as those techniques get developed.
Another question from the audience. The tissue environment and cellular network inside a body is vast and still not completely understood. How would using in vitro techniques, example 3D culture, will be useful to monitor the overall effect of the drug in the body, since it's hard to replicate the exact cell conditions in vitro?
Yeah, and I think that was the exact point that I was trying to make, and that's an excellent observation. In that it will be, in my opinion, very difficult to eliminate all animal testing. But I think we can use some of these alternatives to drastically reduce the testing and also to design more focused animal experiments so that we can get more information out of fewer animals, so that we can have more confidence in the data as being relevant to the human situation. So there are certainly challenges that won't be overcome, certainly in my lifetime, but it the potential is there. Smarter people than I can figure that out.
And maybe another question along these lines. The CIVMs are not well validated for translation to humans, such that we could eliminate some or all animal studies. Further, there are dozens of models that will require validation, and no single company university can complete a single validation. Obviously very costly, need broad compounds, case studies. How do we as an industry, academia, try to validate these models to a level that we can begin to reduce or eliminate animal use?
I think what we're seeing here is a lot of cross-company collaborations in the form of consortia with IQ and ICH and etc. And I think particularly with the organs on a chip, companies need to work together to validate and develop a kind of standardization around the different technologies that are used. We need to explore how predictive they are, looking at in vitro data, organ on a chip data, in silico data, in vivo data with known compounds, and also obviously compounds of interest within the particular project. So there is quite an extensive validation exercise that is required to get to where we are with some of the in vitro techniques, and it's going to be a lot of effort, but companies will need to kind of work together to really generate consensus and standardization here.
And I would add one more thing to that. I think it's really important that regulators are part of that process. So I think we need an ICH-like effort around some of these alternative models where we get the key stakeholders together and discuss the utility, the limitations, what we need to do to have confidence in them. And, again, I hope that that is something that that happens, and because I think that it will really drive the use of these alternatives forward much more quickly than if it's done piecemeal.
One last related question before we go to toxicology, and that is there a trial to translate the results from organ on a chip studies into human pharmacokinetic predictions for new drug candidates?
Do you know what is meant by a trial there?
I don't. I'll just read it one more time. Is there a trial to translate the results from organ on a chip studies?
Well, I think what we've seen in the past with in vitro techniques is that many different companies and academics have looked at trying to explore how in vitro data predicts in vivo data across projects and across compounds and across the industry. And I'm aware of many different consortium around organs on a chip, and I'm sure all of those different groups are looking at how well data generated from those organ on a chip technologies are predicting in vivo data. So I do think those kinds of efforts are going on. I'm not sure I completely interpreted the question correctly there, but hopefully that goes some way to answering it.
Thanks, Hannah. And Jim, let's go to the technology perspective. It would be great. What are if you could kind of focus a little bit on what are the current challenges associated with the use of animals in toxicology and other non-clinical studies? And a follow-up, how are these challenges currently being met?
Okay. Well, I think the biggest challenge today with the use of animals in toxicology and other non-clinical studies is animal supply, particularly of large animals. The supply chain for non-human primates was disrupted by the pandemic, such that the availability of animals became very scarce, and to the point of non-human primates became very scarce, to the point that in February of 2002, FDA issued a guidance document for mitigating that supply constraint. And then drug developers rightly turned to other species such as dog and mini pig. Of course, that increased the demand for those species, which was also impacted by the closure of one of the major suppliers of research dogs in 2002. So it's become more difficult to obtain those species for non-clinical studies. And of course, with the high demand and low supply, the cost of conducting these non-clinical studies in large animals has increased dramatically, in some cases by 50 to 100%, simply based on the increase in animal costs. So how are sponsors dealing with this? And I think, and we certainly encourage sponsors to do this, but we're critically evaluating study designs to reduce the number of animals to the extent possible, while still maintaining a valid study that we can interpret appropriately. So as an example, it may be possible in some early exploratory studies to compare the data in drug-treated animals to a historical control database, rather than include concurrent controls in those particular studies. Most CROs have very extensive historical control databases that you can match by animal age and supplier, and there are some confidence intervals around those mean values. And for early exploratory studies, you can compare those to the historical data rather than have to include controls in the actual study. Another technique that people are exploring is to develop more sensitive bioanalytical methods, so that you can reduce the number of animals that you have to include in rodent toxicology studies to determine drug exposure in those studies. So, that is something else that is also being explored and in some cases used. Sponsors are also evaluating the use of alternative large animal species, although there aren't a lot of great options. However, rabbits, which have been historically used to assess potential developmental effects, are now being considered for repeat dose tox studies. And in fact, just last week, there was a session at the annual Society of Toxicology meeting exploring the rabbit as a non-rodent species in tox studies in response to the shortage of other large animal species. Unfortunately, sometimes these challenges can't always be met in a timely manner, and some non-clinical studies have to be delayed until sufficient animals are available. And of course, these delays can impact clinical development and ultimately what we really care about, making drugs available to the patients. So sort of a domino effect that's happening. And so that I would say is the greatest challenge today for the facing animal usage in tox studies. And if I could, I'll just walk. Oh, go ahead, David.
No. Go ahead, Jim.
Oh, I was just going to say I can sort of walk through this cartoon and sort of it's my impression of where we are now and where we can be in the future in terms of animal usage versus alternatives. And what I've displayed here is the relative size of the individual that's providing support for clinical development is really a reflection of the number of animals used in those various phases. So in the discovery space, I think there are alternatives. I think there's still a fair bit of animal usage that can be, we can shift that arrow much greater to the alternative techniques. Phase 1, phase 2 clinical development, where there are a lot of animals that are being used to support those phases of development, and very few alternatives are being used. I think we can get to the point in the future where maybe that's 50-50. To support phase 3, those types of studies, non-clinical studies that are done are typically what I refer to as the labeling studies. So those are the studies that will inform certain sections of the package insert so that the patients and their physicians can make informed risk-benefit decisions. And right now, those studies are heavily weighted toward the use of animals with very few alternatives. But I think there is a possibility to be able to move that needle a little bit. I don't know that it'll ever be 50-50 or favor alternatives. But one of the things that ICH did recently is they amended one of their guidelines to basically allow sponsors to present a scientific argument as to why they don't have to do a 2-year rodent carcinogenicity study. And the regulatory authorities around the world got together. They did some pilots. They collected some information and the so-called carcinogenicity risk assessment and compared it to the actual results of CARC studies to gain some confidence that it may be possible to, in certain circumstances, eliminate that 2-year rat study, which typically involves, 500 to 600 animals and takes 3 years and $3 million to complete. So obviously, there's a potential for big savings there. And then there is a little bit of support for registration and post-approval. And I would say, it's probably about 50-50 now. And again, I think we can move that needle more toward alternatives.
So we have a little more than 5 minutes left, and we still have a number of questions. So I'm going to do a quick lightning round and see how many of these we can get through. So I'm going to ask these quickly, and Hannah, Jim, if you want to take any of these, feel free. So first question, are you aware of any FIH studies already initiated with preclinical data generated with alternative methods to animal testing? If any, quote, surprising safety issues arise from human data?
I'm aware of at least one situation where there is, based on the target, which was only expressed in humans, animal testing wasn't required. So in that instance, sponsors had to provide information on what was known about the biology of the target and what potential um, hazards could be associated with modulation of that target. And then that required different safety monitoring parameters in phase 1 that were specific to that target and how it was modulated. But I would say that that's an extremely rare circumstance. I'm also aware of some instances where companies have been developing deuterated drugs that contain deuterium atoms instead of hydrogen. And so what they've been able to do in those cases is do some very limited pharmacokinetic modeling, or perhaps limited, in vivo PK studies to demonstrate that there are no differences in the deuterated drug versus the previously approved drug, other than positive pharmacokinetic advantages. So I have seen situations where that's been the case, but again, relatively rare. One of the things
Okay.
that the Act, one of the things that the Act did point out is that if you are developing a biosimilar, you no longer have to do in vivo PK testing before you can move into human with that biosimilar.
All right. Next question. Are there some indications where animal testing might become not necessary sooner than others?
Well, I can think of an example several years ago where we worked on an impurity qualification issue for a client, and we put together a very scientifically based package based on what was known about the target biology, what was known about the active ingredient, and we did some testing, a lot of in vitro testing, and we submitted that to FDA. And they came back to us and said, well, this is very, very nice data. It's very supportive of what you're doing, but you have to do the animal studies anyway. And I'm hoping now with the advent of this act, where we may be able to go back and convince them, again, with good science, that we can forego doing those types of in vivo impurity qualification studies. So,
And I think
sorry, Hannah.
I think in the pharmacokinetic and pharmacodynamic space, particularly in areas where we have a good understanding around how pharmacology links to efficacy, for example, in the antiviral space, I think those are the kind of areas where we're less likely to need animal testing, at least to understand the efficacy component of the molecule.
And then another question, safety boards, internal, and IRBs are also key stakeholders. How do we get alignment to eliminate reduced animal work? Would they support a human clinical study with limited animal data?
I think if I go back to the example of a deuterated compound, and these particular compounds were actually developed in Australia, where IRBs are key decision makers for phase 1 studies. And, sponsors have been able to go in with very limited animal data, primarily around pharmacokinetic behavior, without any toxicity information, and have been able to initiate FIH programs. Again, that's just a very specific example. I think in general, if people are going to follow even IRBs are going to follow in general, the recommendations of ICHM 3R2 with respect to what studies need to be done to support FIH. And until some of these alternate methods are validated, I think it's going to continue to be the case that we're still going to have to to do these types of studies, certainly in the near term, and potentially for some time.
Well, that brings us to the top of the hour. And Jim, Hannah, I do want to thank you for your time in this informative discussion. Certainly appreciate your expertise. To our audience, on behalf of Nasdaq and Certara, thank you to everyone who has joined today's webinar, and have a great rest of your day. Thank you so much.
Thank you.
Thank you.